All right, we're on the air. So let's check the YouTube. I'm pretty sure you made I know. You made that version. Is that sound on from the angle?
That's me.
You're on mute. Did you check that?
Yeah, just doing the circular thing.
Are you saying? Yes.
Yeah.
No. You got freestyle already ready for that.
Yeah, that basic question.
Your day will come. 20 years from now. You'll be standing here. Actually, not 20. 10. Shane, you can still hear us, right?
Yes, I can.
Awesome.
YouTube thing is a little delayed on my phone, but it's up.
Yeah.
Larry, you only want something I don't know. I'm not like a Halloween. Oh, you're right. But I don't agree with like spending your money on a costume that you can't outwear. Like I don't know. It depends on what it is. Like, what would you wear next Halloween? So I'm not. But like,
You need like public day.
no, yeah, I agree. I found a difference word. I'm like, okay, I'm a con, but no. I'm not legit.
First thing, like to make sure, you know, that's his level. That's
You're ready. You don't get to solve. I'm not sure what. Okay.
Yeah, before we made the pool house.
Oh, okay. You, that embarrassed you.
Oh, yes.
Okay, so this is what happened. continue with the that we use for not trying to contact. You say it? So therefore,
Yeah, I know. Yeah, I mean, that's how I basically live in the rest of this is yeah.
Got it one time. Chill. Sasha said I should say scatter one time. What? You gonna do it? You better.
Are they going back to like the lab? For the private portion, it'll be here. Like once you finish, where you go? Everyone leaves and does whatever. Yeah, I'm still I don't know. I'll text you.
Sure. 175. 175.
You guys okay? Ed's in the back.
Have you met my mom? Okay, she didn't remember it.
Yes, sir. Afterwards, there will be a city of the presentation. There will be a link. We're streaming it, so everybody who can't be here can watch it online. I'm still going to take one or two.
I guess it's like.
Got it. I've got to go to that.
Yeah, I'll text you.
Shane's there. He's there? Yeah, I have requested videos to record this. Hi. Are you?
Yes.
And so will it, when we go to the closed session, it's going to send me a text and I'll put her back in and
So it's on. Just call me. Just go to the missed call.
Tony, stand up front so you can take a picture of you. So you can take a picture of you.
Yeah, we have.
Is this for you? That's for Amadou? Where's Amadou? Yeah, put that.
Okay, yeah, I'll be around. Oh, I'm definitely
Huh? She's trying to find parking. She's gonna park. She's gonna be here.
Dude, you have to read. Ready? I mean, ready? I don't know. It's hard to get work, but it's like
Feeling that we're cooking this one.
All right, so I suppose we can go ahead and try to get started. As you'll hear, Tony's kind of a stickler for punctuality, so I don't want to be the cause of him being any later. So, I think that thesis defenses are always a bit, bittersweet, bitter in the sense that, you develop and cultivate a relationship with someone and hate to see them go, but sweet in the sense that you can acknowledge some of the things that they have accomplished. I think, with Tony, I guess about 5 years ago, I decided to do this experiment. I'm an experimentalist. I decided to do this experiment where, I would show the world that you could put, a Nigerian and a Ghanaian together in the same bag, and there could be cooperation. But I had this significant constraint. You had to put the Ghanaian in charge and see what would happen, right? So, would it be kind of a clash of two proud cultures, or would it really be characterized by cooperation and incredible productivity, right? Which is it? And the answer, yes. And a clash of strong wills in deciding on the best and most productive direction to pursue, or would it kind of be a model of cooperation and humility? And the answer is yes. So I think with what I could say so many things, but I'm not going to take up too much of his Tony's slide here. But it's one of the things that I think is emblematic of the kind of relationship we've had and how that's impacted his PhD thesis, is that he has to be, if not the, or maybe one of only a handful of PhD students in the history of getting PhDs that could get a first author paper in PNAS, which for those of you who are non-scientists is a very, very highly competitive and prestigious journal that has nothing to do with the subject of his PhD thesis, right? And part of the reason why that's only a slight exaggeration, which you'll hear about, but part of the reason that's true is about 3 years in, even though when I met Tony and tried to convince him to do a PhD and not a master's, one of his primary complaints was, hey, I've got the next 5 years of my life kind of planned out, and I don't think I'm gonna have time to do this PhD thing. So I said, no, we can get you finished with a PhD really, really quickly. And he said, okay, I'll trust you this much, but I gotta get done in a certain period of time. Well, 3 years in, when the completion of his PhD was on the horizon, we both got the idea that sickle cell disease was a topic that was worthy of our attention and to be a focal point for our research. It was completely different than what he had been doing, and it would be like starting over 3 years into a PhD for someone who is extremely goal-oriented. And before we set out on it, I said, Tony, look, if you make this decision, it could mean, adding significant years to your PhD. And, I know how much that means to you. So I want to make sure that you're going to be driven by a passion to really address this problem before we make this decision. Why don't you go and you think about it? And he came back, he thought about it, and he committed himself to a direction in doing sickle cell research. And what I think he was able to accomplish in a very short period of time, having pivoted off his original project, is nothing short of spectacular, or so I'm not unbiased. So with that, I'll hand the floor over to Tony for his defense.
Thank you for that energetic, partially true, introduction. So there was a seat in the front. I guess there isn't anymore. Sorry about you guys. So today I'm going to discuss what I've dedicated the past few years of my life to. Very passionate about the topic. And I know there are a lot of big words on this slide, but my goal at the end of the presentation is to convince you that sphingolipid metabolism is important in sickle cell disease, and it actually contributes to inflammation and some of the pathogenesis mediated by these things called microparticles. I also just went to the dollar store this morning and bought this laser pointer, which just broke. So I unfortunately don't have a pointer, but I'll try to use the mouse and my finger to navigate it. So sickle cell disease is, like I said, a topic that's very passionate to me. I have family members with the disease, and I also have a trait myself. It's a hereditary blood disorder that's caused by a single point mutation in the gene for hemoglobin. And basically what happens is the hemoglobin monomers form, they get really, really sticky, like skin. They get sticky in the disease state, the mutated state, and they form these polymers that polymerize and distort the red blood cell membrane. And that forms this canonical sickle shape. These red blood cells themselves are more adhesive and can stick to the endothelium that lines blood vessels. They also can completely block off small capillaries and blood vessels that you're seeing in the bottom right, which leads to a phenomenon known as vaso-occlusion, or the vaso-occlusive crisis. It's not only very painful, it significantly cuts off blood, oxygen, and nutrient transport to downstream tissues, and can lead to tissue and organ damage, significant vascular complications, such as stroke, peripheral arterial disease, and even death. Now, why is sickle cell disease important? So it's important to study therapies for disease because there is no cure that's used in humans. And the economics of sickle cell disease are significant. Their annual healthcare costs that exceed $1 billion in the United States. And as you can see to the left, due to there not being cost-effective therapies to treat complications of the disease,
children with the disease can spend as much as $15,000 annually just in treatment. So another thing that I haven't mentioned yet that you'll begin to appreciate is sickle cell disease is a very, very complex disease. It's not just the red blood cells that are damaged. They can signal to other cells in circulation, such as monocytes and neutrophils that are inflammatory and can propagate some of the disease processes. So we sought out to really understand the interaction with these sickled red blood cells and inflammatory cells like monocytes and design therapies to reduce inflammation, specifically vascular inflammation in the disease state. So here's a diagram showing how the vaso-occlusive crisis generally works. So it happens in multiple phases, but the four most important ones start with RBC adhesion. So these sickled red blood cells are more adhesive. They themselves can adhere to endothelial cells lining the endothelium. They're also more prone to hemolysis, which is basically bursting or lysing of the red blood cell, which not only damages the cell, but it releases the intracellular contents, which oftentimes are pro-inflammatory, like nitric oxide and ET1. These pro-inflammatory cytokines or molecules and others can activate other cells, like I mentioned, monocytes and neutrophils, leading to further adhesion in these blood vessels and downstream activation of platelets, neoenteral and smooth muscle cell proliferation and thickening, leads to ultimate vaso-occlusion, which completely walls off small capillaries and vessels. Now, if we take a careful look at the red blood cell membrane, you can really appreciate its ability to concentrate lipids on either part of it. So all cells are separate their intracellular components from the plasma or extracellular space by this plasma membrane that has two layers of lipids. The outer leaflet, which is exposed directly to the plasma, has a specific kind of profile. There's a concentration of specific lipids that are abundant on the outer leaflet, and other lipids are existent on the inner leaflet. In sickle cell disease, for example, phosphatidylserine, this red lipid, has been shown to be translocated to the outer leaflet, and that serves as a signal for clearing damaged cells. So when phosphatidylserine is exposed on the outer leaflet, macrophages and other inflammatory or phagocytic cells can engulf and clear these damaged RBCs from circulation. Our lab is really interested in sphingolipids. What's a sphingolipid? Here's one of them, sphingomyelin. As you can see, it's significantly concentrated on the outer leaflet. It's called a sphingolipid because it has a sphingoid backbone, or backbone of a long carbon chain, with a variable chain. In this case, it's a fatty acid that has 18 carbons, or 18 carbon long fatty acid, and it has a large polar head that kind of sits in the plasma membrane. So sphingolipids exist, or are called sphingolipids, because they have the long sphingoid or carbon backbone and a large polar head. Now, they play structural roles in the red blood cell membrane. The sphingomyelin, again, it's concentrated on the outer leaflet, but there are other lipids, sphingolipids that exist, like sphingosine-1-phosphate, that play bioactive roles. Now, I had mentioned some of the earlier work that we did before I transferred over here from the University of Virginia to Georgia Tech, and most of that work looked at the role of sphingosine-1-phosphate in the recruitment of a distinct subset of monocytes known as the anti-inflammatory or pro-regenerative monocyte. So these lipids don't just sit in the red blood cell membrane and contribute to their integrity and stability. They actually signal and interact with other cells in circulation. And we became really, really interested in that balance between structural integrity and bioactive signaling in sickle cell. So just to give a brief overview of sphingolipid metabolism, again, we have sphingosine-1-phosphate. We have a long sphingoid or carbon backbone with varying degrees of saturation. These long chain bases are called long chain bases because of the long carbon chain. And they have different polar heads as well as different degrees of saturation or double bonds. We're looking at sphingosine-1-phosphate here. Sphingomyelin, the structure is different because of this variable fatty acid group that can have different lengths and also different degrees of saturation. But altogether, there are many different classes of these lipids, and also many different lipids within the class that exist in red blood cell membranes and all cell membranes. So we became really interested in the interaction between these lipids, specifically how the enzymes might be modulated or dysregulated in sickle cell disease. So again, these are the lipids outlined in blue are the complex phospholipids that have variable fatty acid chains and the long chain backbone. And the four in red are the four long chain bases that just have that one long chain. So we focused primarily on this reaction here for the majority of this work. And hopefully by the end of the talk, you'll appreciate why. But this is one of the first papers that we looked at that connected inflammation and sphingolipid signal in the metabolism. So basically what these authors found was that if you inject these two cytokines, interleukin 1 beta and TNF alpha, you can drive inflammation or the production or secretion of acid sphingomyelase, the enzyme that converts sphingomyelin into ceramide in circulation. And this happened in a dose and time dependent fashion. So sickle cell disease, I mentioned, is a disease that's characterized by systemic inflammation. So we expected to see similar changes in enzymes that mediate sphingolipid metabolism in sickle cell disease. Additionally, a more recent paper looked at the metabolism mediated by a different form of acid sphingomyelase called neutral sphingomyelase. This enzyme exists in red blood cells, and they found that if you introduce forces in the red blood cell membrane, and they did this by throwing red blood cells in hypotonic or hypertonic solutions, which would cause them to expand or contract, you introduce forces in the red blood cell membrane, and this neutral sphingomyelinase, which again is the enzyme that converts mediates this reaction, is actually activated. Now, why is that important in the context of sickle cell disease? Here's a scanning electron microscopy image that I took of a sickled RBC. As you can see, it doesn't look like a red blood cell. It's not spherical, it's not biconcave. It has significant bending and folds in its membrane caused by the polymerization of hemoglobin. And you can imagine significant forces being introduced in this membrane that have the potential to activate enzymes like neutral sphingomyelase and others that are mechanosensitive. So lastly, we wanted to look at the functional consequences of sphingomyelinase activation before we got into the majority of my work. And we found this paper that treated red blood cells, and here you can see that they're very round and biconcave. You can see two different planes. But when they treated these cells with acid sphingomyelinase derived from bacterial cells, they saw that the round biconcave shape of the red blood cells was lost. But more interestingly, they saw that these membrane-derived particles, membrane-derived lipid vesicles, what we call microparticles, were enriched in the intracellular space. This was the first group that looked at production of what we call microparticles by red blood cells mediated by sphingomyelins. Now, why are microparticles bad in the context of sickle cell? We went to the literature, and time again, we found studies that associated sphingomyelinase or microparticle expression with inflammation, atherosclerosis, and other signs of vascular inflammation, not unique to sickle cell, but in many different models in both mouse and humans. So the main story here is that microparticles which exist in sickle cell disease are pro-inflammatory and can enhance vaso-occlusion in the disease. So here's just a schematic of the red blood cell membrane. We just discussed briefly the plasma membrane that has the outer and inner leaflet. What we knew to this point was that sphingolipids exist in the plasma membrane. Here I'm showing 4: sphingomyelin, ceramide, sphingosine, and S1P. Enzymes such as acid sphingomyelinase and neutral sphingomyelinase also can associate with the membrane and convert sphingomyelin into ceramide, which can further be converted into into sphingosine with ceramidase and bioactive sphingosine 1-phosphate or S1P with sphingosine kinase. Downstream of acid sphingomyelinase activity, we know that membrane-derived exosomes or microparticles can be produced, and these don't just sit in the red blood cell. If the red blood cell lyses or if they're actively secreted, they can be introduced into plasma where they can interact with other cells in circulation. So our overlying question was, is sphingolipid metabolism and microparticle production altered in sickle cell disease? And our hypothesis was that pro-inflammatory microparticle production is a consequence of sphingolipid metabolism and contributes to vascular inflammation through monocyte activation. To test this hypothesis, I designed 3 aims. The first, to explore sphingolipid metabolism and subsequent microparticle production and characterization in sickle cell disease. The second, to assess the effects of these microparticles on inflammatory cells, such as monocytes. And the third, to interrogate the pharmacological or therapeutic modulation of sphingolipid metabolism in a mouse model of disease. So in aim 1, I sought to evaluate the changes in sphingolipid metabolism, the resulting changes in the red blood cell lipid state, sphingolipid state, and characterize red blood cell derived microparticles that exist in sickle cell disease. To accomplish this, we collaborated with a group here at Georgia Tech, the Platt Lab, and I have to thank you guys a little bit later about for allowing us to get build this collaboration, this relationship with the Sickle Foundation of Georgia. But this is a group that basically allowed us to collect blood samples from donors that had sickle cell disease or didn't. The majority of the talk I'll refer to the sickle donors as SS and the normal or non-sickle as AA. But when we got these whole blood donations, we were able to separate them into 3 different components using a Phi-called density centrifugation method. I don't know if you guys got a chance to grab the candy in the back, but there's candy back there that has plasma, white blood cells, and red blood cells. It's exactly like my samples do. They just taste a little bit better. But the plasma has most of the proteins in the blood. The buffy coat layer has your white blood cells and platelets, and the packed red blood cell layer has the red blood cells and microparticles. So we interrogated these 3 different layers and wanted to see if we could detect changes in sphingolipid metabolism. First, we looked at neutral sphingomyelinase, which again is the enzyme that was shown to be activated upon bending stresses. And we didn't see a significant increase in the activity of that enzyme in the plasma, but in the red blood cells, we saw a slight but significant increase in its activity. What was interesting though was that the magnitude of the activity was relatively small, so we decided to look at the other isoform of sphingomyelinase, acid sphingomyelinase, and found that this enzyme was significantly increased in its activity in both the plasma and the red blood cells of people living with sickle cell disease. So activity can be increased because of an increase in enzyme expression or its abundance, or a conformation change in the enzyme that increases the activity of the individual enzyme. So we looked at expression. We didn't see significant increases or changes in enzyme expression, which suggested that the actual enzyme was activated. To test this hypothesis, we used Purcell density centrifugation, which is a technique that allows us to separate red blood cells or any cells based on their density. More dense cells will lie at the bottom of the centrifuge tube after we spin down, and the less dense cells will be at the top. So density is a way to measure or harvest cells that are damaged or sickled, because as they sickle, you have the same mass, a lower volume, and the density increases. So the highest density cells in these sickle donors are irreversibly sickled red blood cells. We tested the acid sphingomyelinase activity in each of these fractions and saw that as the cells sickled or increased their density, they also increased their their activation of sphingomyelinase, suggesting that sickling induces activation of the enzyme that we're studying. And we also wanted to look at enzymes that were downstream of sphingomyelinase activity. We looked at ceramidase, which again converts ceramide into sphingosine, and saw that it was significantly increased in the plasma, suggesting it could be secreted in the disease state. Sphingosine kinase, which takes sphingosine and forms S1P. One isoform, sphingosine kinase 2, was significantly increased in red blood cells in sickle cell disease. And again, these red blood cells aren't making much. They don't have the ability to transcribe their own protein. So most of the changes that we're observing, we hypothesized, came from plasma. So we looked at the plasma and found that just like ceramide or ceramidase, sphingosine from people living with sickle cell disease, and was detected at higher levels there. Now, just to remind you of the metabolic network, we have sphingomyelin, the most abundant sphingolipid in the red blood cell membrane, which is converted to ceramide, sphingosine, and S1P by sphingomyelinase, ceramidase, and sphingosine kinase, respectively. All these enzymes were increased in their activation or expression, so we expected to see increases in sphingosine and S1P in the red blood cells. So we looked in the whole blood, plasma, and red blood cells themselves, and found that consistently in sickle cell disease, we see an increase in both, a significant increase in sphingosine and S1P. So microparticles. Others have attributed sphingolipid metabolism with exosome and microparticle formation, so we wanted to see if microparticles were increased in our samples as well. We first looked at the plasma, and we relied on flow cytometry, which allowed us to look at the size distribution as well as the expression of phosphatidylserine. We used Nextin-5, which binds to phosphatidylserine on the red blood cell membrane, or the microparticle membrane, to detect these particles by flow. We didn't see a significant increase of their presence in the plasma, but when we looked at the red blood cell fraction, we saw a significant increase in microparticle production. Now, what do these microparticles look like? Hopefully you can see the video at the bottom left, but we stain these red blood cells, sickled red blood cells, with lipophilic dyes, di-I and di-O. Di-O is a molecule that binds preferentially to the inner leaflet. So wherever you see this punctate green staining, that's di-O expression on microparticles. So we could actually see these cells forming microparticles. And when we looked at them using transmission electron microscopy, we looked at 3 different size distributions that detected that we harvested using 3 different centrifugation steps. And as you can see, they range from about 200 to 400 nanometers in diameter. And our immediate question was, do these microparticles also have changes in sphingolipid metabolism? Or do they express the enzymes that we're interested in? So sphingomyelinase actually contributes to the production of them. We thought that the enzyme itself might be encapsulated in the microparticles themselves. So we looked at sphingomyelinase activity in the microparticles, and we didn't see much expression or activity in AA microparticles. But we did see similar distributions or modifications in terms of the lipid state. We saw an increase in the ceramide to sphingomyelin ratio, which is consistent with sphingomyelinase being activated. And we also saw a significant increase in sphingosine and S1P, the 2 lipids that are downstream of sphingomyelinase activity, and sickled microparticles. So to this point, we focused a lot of our attention on sphingosine and S1P, which we know is bioactive and immunomodulatory. We wanted to look at different lipids that we knew existed in the metabolic network. So we analyzed 86 different sphingolipids, and we were able to detect them using HPLC, high performance liquid chromatography, and tandem mass spec, which is basically a technique that's used to quantify molecules. So if you look at the structure of the complex lipids, I mentioned that there's this stable long chain base, but this variable side chain, fatty acid chain, can have lipids that are 13 to 26 carbon longs and have different numbers of double bonds along their structure. So with sphingomyelin, for example, it's not just one molecule. Some of them are 13 carbons long, some are 16, and the numbers behind the colons here are basically the number of double bonds that exist in the structure. So we were able to look at all of these molecules in our different classes of complex and long-chain based sphingolipids and detect significant differences, or look for significant differences in expression. So we revealed we performed a volcano analysis, which basically takes tons of measurements between 2 samples and detects at a specific confidence level specific molecules that are significantly altered in their expression between the 2 samples. So in this case, we found 8 lipids that were significantly different. We didn't know at this point if they were increased or decreased in sickle cell disease. And we looked carefully at those 8 lipids and found that, interestingly, 4 of them were a long chain basis. So this is sphingosine, this is sphingosine, which is sphingosine with a double bond, or without one, excuse me, sphingosine 1-phosphate, and sphingosine 1-phosphate. So all of the long chain bases were consistently elevated in our sickled RBCs as well as the microparticles. The other 4 that were changed were 3 of them were ceramide 16, 18, and 22, and 1 was sphingomyelin, which was actually reduced. So this is very consistent with the increase in acid sphingomyelinase activity. We expect an increase in the product, ceramide, and a reduction in the reactant, sphingomyelin. So it's really interesting to see that we were not only seeing changes in enzyme expression and activity, we're seeing changes that are consistent with these enzymes in the lipid expression in both long chain bases and complex lipids. So now we went back to the lipid families. There are 9 lipid families that we looked at. And we wanted to see if there were any differences in the total amount of, let's say, ceramides or ceramide 1-phosphate between AA and SS red blood cells. So our first observation was that ceramide 1-phosphate was reduced in SS RBCs. I just mentioned this, but all of the long chain bases, sphingosine 1-phosphate, sphingosine 1-phosphate, and sphingosine were significantly increased in both RBCs and microparticles. And ceramide and dihydroceramide, which are immediately downstream of acid sphingomyelinase, were also significantly increased. So to this point, we can go back to the metabolic network and we can say, hey, we've made all these really interesting observations on enzyme activity, enzyme expression, changes in lipid expression, some decrease, some increase. We don't know everything, right? We don't know, for example, whether the flux mediating the conversion of sphingosine to ceramide is different based on the changes that we're observing. So we decided to rely on mathematical modeling to really ask questions about how the reactions or the fluxes were changing between the sphingolipids that exist in this metabolic network. And we also wanted to account for transport between the RBCs and plasma. So to do this, we used what's a modeling approach called flux-based analysis. And basically, this is a simplified version of our metabolic network. You can imagine a network that has 3 different metabolites, in this case A, B, and C, that's mediated by 3 different reactions, V1, V2, and V3. We call these reactions fluxes. So steady state, and I didn't mention this earlier, but all the samples that we've gotten, we're considering them at steady state because the donors hadn't been hospitalized recently. They weren't taking medicine for sickle cell disease. So the measurements that we were making were at their baseline or homeostasis. So steady state, we wouldn't expect the concentration of these metabolites to be changing over time. Therefore, we can add up the fluxes that produce or consume these metabolites and know that they must balance to 0. There's no net production or removal of these levels. We can then use the metabolic architecture to create what's called a stoichiometric matrix, which basically defines all of the metabolites with respect to their individual fluxes. In this case, if we look at metabolite C, it's built by 1 unit of V1, 1 unit of V2, and removed by 1 unit of V3. So if you look across the rows, this stoichiometric matrix tells us what reactions play roles in which metabolites. So again, we're at steady state. We can solve this stoichiometric matrix for all of the fluxes or the reactions that can be combined with it to produce steady state, or S times V equals 0. And we can constrain this by experimentally measured and biologically reasonable fluxes for each of the reactions and transport processes. So before we got to the to running the mathematical model, we refined our metabolic network with reactions that we knew existed in the red blood cells. So not all of the reactions, for example, some of the reverse reactions exist in red blood cells. So we limited it to the reactions that we knew existed. And this resulted in 11 different reactions mediated by the 11 enzymes that are outlined here. Also mentioned that we wanted to account for import and export. So the red blood cell is metabolizing these lipids by itself, but it also is in exchanging these lipids with the plasma. So we considered import or uptake from the plasma and export, spitting into the plasma of all of these lipids, and that resulted in a total of 29 fluxes. So here's the data. These are the 29 different reactions. The model basically predicted that, so this is the fold change in the flux, or the abundance of the flux in SS over AA. The model predicted that there was as much as a 35-fold reduction in one reaction and as much as a 3-fold increase in the flux between metabolites or the transport processes in sickled RBCs. 7 of those fluxes were decreased more than 2-fold. For example, reaction 22 is right here. That's the export of dihydroceramide, which is reduced almost 35-fold. One of those was increased more than 2-fold, and that's sphingosine kinase. What was interesting about this particular enzyme is that a recent paper in JCI, the Journal of Clinical Investigation, found that sphingosine kinase is significantly activated in sickled RBCs and contributes to sickling. So we were really excited to see that the model was predicting things that were similar to stuff that we had observed, as well as others in the literature. So to this point, I've talked a lot about the production of microparticles being mediated by sphingomyelinase, right? So if we know that sphingomyelinase is playing a role in the production of these particles, why do we care about anything else happening in the metabolic network? That's because the flux in this reaction actually isn't that different between AA and SS RBCs. There's about a 10% increase in the flux mediated in this reaction. When we looked at the magnitude of the velocities, these fluxes basically are quantified in picomole of the lipid produced per minute, normalized to the amount of protein in the red blood cells. Reaction 10 is orders of magnitude faster than any other reaction in the metabolic network. So basically what that means is that even a sphingomyelin can be converted to ceramide at this rate, about 200 picomoles per minute. The rest of the metabolic network can't handle that flux. So instead of all of that product, all of ceramide being crammed into the metabolic network to be further processed, it's exported. So the cell handles this by exporting as opposed to importing these lipids. So if we truly wanted to design a therapy that modulated the expression of lipids and not just the production of microparticles, we might want to look at reactions that are much slower, rate-limiting reactions that we can actually control, such as reaction 8 and 1 that happen at a much slower rate than sphingolipids. So back to our schematic of the red blood cell, AIM-1 accomplished a lot. It told us that membrane stresses can activate acid sphingomyelinase in the red blood cell membrane. Acid sphingomyelinase can convert sphingomyelin into ceramide, which can further be converted into sphingosine and S1P. These microparticles can be produced at regions of acid sphingomyelinase activity, and they're sphingolipid rich. They also express they express the same changes in sphingolipids expression as the red blood cells themselves. And these microparticles can be released into circulation where they can interact with other cells. Now back to our cascade of vasoocclusion. We're really interested in this step here, the third step. And that's when inflammatory cells in circulation can become activated by something in the sickle disease state. We really wanted to see if the microparticles that are being exported from these red blood cells could potentially modulate some of these inflammatory processes. So to answer that question, I designed AIM-2, which was to evaluate the interaction of the red blood cell-derived microparticles, specifically the sickled red blood cell-derived microparticles, with inflammatory cells like monocytes, and the resulting changes in behavior, which are measured by endothelial cell adhesion, cytokine production, and genetic modification, which would all contribute to vasoocclusion in the disease state. So our first question was, is adhesion different? So that schematic that I showed a couple slides back showed that the monocytes can become activated and hyperadhesive to endothelial cells. So we took human umbilical cord vascular endothelial cells and cultured them in confluence in 96 well plate. So you take your endothelial cells and make a confluent layer, and then you can take monocytes. In this case, we used THP1 monocytes, which is a human monocyte cell line. And we treated the monocytes either with S1P, or bioactive lipid, or AA or SS plasma. And we found that only with S1P and SS plasma incubation did we see significant increase in the adhesion of these treated monocytes to endothelial cells. So S1P itself could be modulating adhesion, but so could something else in the sickle plasma. We also wanted to see if the direct co-incubation, or the direct interaction between sickled RBCs and THP1 monocytes could enhance adhesion. So we co-incubated the THP1 monocytes at a ratio of 10:1 with SS or AA disease, benign disease red blood cells, and found that the SS RBCs significantly enhanced the adhesion of these monocytes to endothelial cells. So here we have three different mechanisms, potential mechanisms: S1P, the lipid, something in sickle plasma, or the red blood cells themselves. S1P is the one thing that could potentially be in all of these, that's significantly enhancing the adhesion of inflammatory cells to endothelial cells. Now back to microparticles. Do microparticles do the same thing? The answer is yes. I'll remind you that microparticles also have sphingolipids. S1P significantly increased in SS microparticles. But what's interesting about this result is that the AA, or the non-disease microparticles, didn't significantly enhance monocyte adhesion, but the SS microparticles did. So there's something in these sickled microparticles. It might be a lipid, it might be a protein, or some signaling cascade that's enhancing the adhesion of monocytes to endothelial cells. It's not happening with normal RBCs. So we found this paper. This is a group, Zanati's the first author, that's been publishing in the sickle cell area for years. They published two papers in 2008 and 2012 that built this mechanism. Basically what they showed in red blood cells, they didn't look at microparticles at all. They showed in red blood cells that this kinase, ERK1/2, was significantly activated, constitutively phosphorylated in sickled red blood cells. And downstream of that, they found that ICAM4, which is an adhesion receptor expressed by red blood cells, was activated. So they basically discovered that the activation of ERK1/2 mediates the activation of ICAM4 and the subsequent binding and activation to and of monocytes. So we wanted to see if this signaling mechanism had existed in microparticles as well. To this point, no one had looked at signaling in microparticles in the context of sickle cell disease, so we wanted to see if ERK was present and if it was signaling. First, we looked at ICAM4, and I'll remind you guys that these microparticles are pinched off from the membrane, so they're not different not significantly different from the red blood cells themselves. They're just domains in the red blood cell membrane that have lipids and proteins that form these lipid vesicles that are smaller and can be secreted. So we expected to see ICAM4 expression, which was there. And we also saw significant phosphorylation or activation of this kinase that exists in sickle RBCs in the SS microparticles, but not the AA microparticles. They're significantly increased in the phosphorylation. So we're still following up this work, but we believe that the ERK signaling cascade could potentially be mediating the adhesion to and activation of monocytes in sickle cell disease that only happens in sickle microparticles. So now we know that the microparticles enhance the monocyte adhesion, but we don't know how these particles are getting into the cell and being processed by the cell. So to answer this question, we took a packed red blood cell layer, ultracentrifuge to harvest our microparticles, stained with the fluorescent dye, CFSE, and then polarized THP1 monocytes to adherent macrophages, mature cells, and incubated these monocytes with macrophages. What we were looking at was internalization as well as modulation of protein or cytokine production and secretion. So first we found, I hope these videos work, we found that these particles could be internalized as whole particles. You can see in many cases around this cell, there's puntate green staining suggesting that the particles are internalized as particles and then subsequently dispersed throughout the cytoplasm. This happened with both AA and SS microparticles. So it's not like we're only seeing results with SS microparticles because they're the only types that are being internalized. The AA microparticles are also being internalized as rapidly as 30 minutes after incubation and are dispersed throughout the cell, cell cytoplasm. Now cytokines, I mentioned this briefly in the beginning, but inflammation is pathognomonic in sickle cell disease, and there are many cytokines that are consistently associated with inflammation as well as vaso-occlusion. 3 of those are interleukin-6, tumor necrosis factor alpha, or TNF-alpha, and interleukin-1 beta. So there are tons of studies out there that have taken blood and serum measurements from people going through painful crises and vaso-occlusion, and have found that these 3 cytokines are consistently upregulated in the plasma. So when we treated the THP1 macrophages with our SS microparticles, we saw that there was an increase in the production and the secretion measured by the intracellular lysate or the extracellular conditioned media of these 3 cytokines, the THP1 monocytes. So we thought, great, we're building a mechanism where we know that these microparticles, and we're finding that these microparticles enhance the production and secretion of cytokines that are already associated with crisis and disease state. So we thought it was great. We went to publish this, and the first comment from the reviewer was that you need to show this in primary cells. You're using a cell line, and you have no idea if this is going to happen in actual monocytes derived from donors. So we did just that. We took AA PBMCs, peripheral blood mononuclear cells, which are for all intents and purposes monocytes. We treated them with SS microparticles. So you have a normal monocyte from a non-diseased donor and an abnormal microparticle from a diseased donor. Treated these normal monocytes with the microparticles and saw a significant increase consistently with the production and secretion of all of these cytokines. So this suggests that if you remove a monocyte from its normal non-diseased environment and treat it with a microparticle from that environment, you can kind of mimic some of these responses or changes in cytokine expression. With the help of our collaborators, Dr. Gibson's lab, we wanted to look at transcriptional modifications in these peripheral blood mononuclear cells. So we centrifuged our buffy coat and harvested our PBMCs from AA or SS donors. We used single cell flow activated cell sorting to sort individual monocytes into a 96-well plate. So that sorts 1 cell into each of these wells. And then we designed a primer set. Now, primers are basically pieces of DNA that we use to amplify RNA and detect how much RNA expression exists in cells. So we designed primers against 96 different genes, 45 of which were Sphingolipid metabolizing enzymes. We looked at non-classical and classical markers of monocyte activation. We looked at S1P receptors, cathepsins, adhesion molecules, and matrix metalloproteinases, or MMPs. We combined these primers, 96 of them, with the monocytes and performed transcriptional analysis to see if there were changes in the expression profile on an mRNA level. Now we're not looking at the protein, we're looking at the step before proteins, mRNA. So there's a lot of data on this side. What I want you to appreciate is the number of measurements that we made and the magnitude of the changes. So again, we're looking at the fold change in mRNA expression of SS PBMCs relative to AA, or non-disease PBMCs. What I'm highlighting in this table are the 5 genes that were most significantly amplified in SS PBMCs. And one of them, interleukin-1 beta, which actually was the third most significantly amplified gene, is the same marker that we've been looking at consistently in vitro and in vivo as a marker of vascular inflammation in sickle cell disease. So it's really exciting to see that we're not only seeing an increase in the protein expression, about a 30-fold increase in SS microparticle treated AA PBMCs, but the SS PBMCs, even in the absence of treating them with microparticles after removing them from their physiological state, there's a similar increase in mRNA expression. We're really interested in sphingolipid metabolism. Changes that we're observing in the red blood cells were being propagated to the monocytes, potentially mediated by microparticles. And we saw this trend here, where there's an increase in ceramidase and a decrease in ceramide synthase. These 2 enzymes mediate opposite reactions, so this suggests that there could be some feedback mechanism going on in sphingolipid metabolism. We also looked at genes that were significantly reduced. Sphingosine kinase 2 was reduced 6-fold in SS-PBMC, suggesting that in this portion of the metabolic network, there would be a net production of sphingosine. Now we also want to look closely at the middle. So we already looked at the 5 most increased and decreased genes, but there's a lot going on in the middle as well. We first looked at receptors, sphingolipid enzymes, and cathepsins. With the receptors, we're really interested in S1P3 because that's a receptor that we've already studied on anti-inflammatory monocytes, and CX3CR1. There's a paper that was published back in 2007 that found that there's a distinct subset of monocytes marked by this CX3CR1 receptor expression that actually calls on vessels and controls for sites of inflammation and can contribute to tissue repair or blood vessel growth where inflamed sites. We found that this receptor was increased 5-fold in SSPBMCs, and S1P3 was increased 2-fold. We also looked at sphingolipid metabolizing enzymes just as a proof, to prove that we're not just seeing transcriptional modification, but we're actually seeing increases in protein expression. There's a 2-fold increase in the mRNA expression of ceramide kinase, and when we looked at immunofluorescent staining, we saw that there was an increase in the expression of that enzyme in the SSPBMCs relative to AA. Lastly, this was a really interesting story. So our collaborators in the Platt Lab published 2 years ago this work showing that these cathepsins, the powerful proteases that can cleave protein members in the extracellular matrix, so collagen, elastin, fibronectin. These cathepsins were found to be induced in endothelial cells when they were co-cultured with sickled monocytes. So if you take PBMCs derived from a sickle cell donor and just co-incubate them with normal endothelial cells, you see a significant increase in the expression of cathepsin K and V. But to this point, no one had looked at the expression of these cathepsins in sickled monocytes. And we found that cathepsin V, one of these, was significantly increased in SS monocytes relative to AA monocytes. So we scratched our heads and we were wondering if the monocytes themselves could be taking in these microparticles, changing their genetic profile, spitting out all of these proteins which are transferred to the endothelial cells, which then begin to degrade and damage the endothelium in sickle cells. To start to answer this question, we looked at again, we treated these SS and AA PBMCs with microparticles in vitro and or in a dish, and measured the expression of, the activity of 2 forms of cathepsin, K and S. So the Platt Lab, they're experts in enzymography. I'm not, but I'll say briefly how this works. They basically have a gel where the substrate for your reaction, in this case, collagen, is embedded in the gel. So when you run your sample through the gel, wherever there's activity, you can run your sample, renature the protein, And if there's the enzyme that mediates that reaction, we would expect the substrate in the gel to be degraded. So these bright white bands are basically showing areas where there's activity of these cathepsins. We looked at untreated M0 macrophages, just derived from our cell line, and found that they express cathepsin K and S activity. Interestingly, when we looked at the AAPBMCs, these are adherent cells, these are not adherent cells derived from our donors. We didn't see much cathepsin S activity. We did see cathepsin S induced in the SSPBMCs. It was about the same level, of cathepsin K activity. When we took these 3 different cell types and treated them with the microparticles, the results were astounding. There was a significant, strong, consistent increase in cathepsin S expression, activity, as well as cathepsin K. Here's the data to the left. When we quantified the fold change in microparticle treated cells over non-microparticle treated cells, there was as much as a 130-fold increase in cathepsin S activity and an 8-fold increase in cathepsin S activity in AA treated or SS treated PBMCs respectively. So we truly believe that these microparticles are internalized by monocytes and are producing all of these inflammatory changes. They're enhancing the adhesion of the monocytes, they're enhancing the expression of cytokines as well as cathepsin expression. So to put this all together, we'll go back to our red blood cell schematic. Now we know that these microparticles don't exist in solitude, they can be internalized by monocytes. They're sphingolipid rich, so they can deliver lipids which are bioactive, like S1P, to the monocytes, which can activate receptors that exist on monocytes, in this case S1P receptors. These receptors can initiate downstream signaling cascades, which include the production and secretion of inflammatory cytokines associated with inflammation in sickle cell disease, as well as the adhesion of the monocytes mediated potentially by ICAM4 and erythrocytes. All right, so our lab is historically a tissue engineering biomaterials lab, so we do a lot of animal work. And at this point, I was itching to get my hands on an animal model. I've done a lot of stuff with cells and proteins, but I wanted to see if some of the things that we were observing were occurring in a mouse model of sickle cell disease, and that was the crux of aim 3. So basically, I'm interrogating whether pharmacological reduction or using drugs to reduce sphingomyelinase activity could do 2 things. Could reduce microparticle production in vivo and could reduce subsequent inflammation in a mouse model of sickle cell disease. So we relied on the Talon mouse model. We got these from our collaborators in the Flat Lab again. And here's a picture of Dr. Chris Patsy at Berkeley. He created this mouse back in 1997. And basically, what they did, what the group did, is they knocked out both forms, or both genes of mouse hemoglobin. There are 2 monomers that exist. There's an alpha and a beta. So they knocked out both alpha and beta hemoglobin, and they knocked in the normal alpha hemoglobin from human and the sickled beta hemoglobin. So they basically created a genetic mouse model that mimicked the sickle cell disease in terms of its degree of sickling and its induction of sickling under hypoxic conditions or low oxygen conditions. So when these mice are breeded with their AS, non-sickled but trait-carrying littermates, half of the offspring have the disease and half don't. So here's just a picture of their spleens so you can appreciate the differences that exist. It's just one measure, one thing that you can look at to confirm that the mice actually do have the disease. Their spleens are significantly enlarged, and that happens because there's spleen dysfunction in sickle cell disease due to the rapid turnover, clearance, and production of cells in the spleen. And your spleen is kind of your reservoir of inflammatory cells. So we created this mouse model where we drew blood from the mice so that we can see what's going on at a baseline. We injected saline, which is just a physiological buffer, or a drug dissolved in saline. 1 hour after injection, we placed mice in a normoxic environment or an 8% oxygen hypoxic environment. And again, upon deoxygenation, the sickled hemoglobin begins to polymerize and induces crisis. And then 24 hours after hypoxic conditioning, we harvested blood and tissue. So we already looked at changes in sphingolipid metabolism, enzyme expression, and activity in human cells, blood, and plasma. We wanted to see if this occurred in a mouse model. So we looked at acid sphingomyelinase activity. What we found was that under normoxic conditions, so even before we induced the vaso-occlusive crisis, there was a significant increase in the activity of acid sphingomyelinase in these mice relative to their trait-carrying litter mates, which are phenotypically normal. But we could induce the activity of this enzyme even further by putting these mice under hypoxic conditions and enhancing the activity of this enzyme. So we sought to look for inhibitors of acid sphingomyelinase that have been used in literature before. And this paper used a panel of tricyclic antidepressants. These are molecules that are used to treat depression. And they have off-site targets on acid sphingomyelinase. So they're effectively functional blockers of enzyme activity by causing them to dissociate from the membrane. So what they found in this mouse model of cystic fibrosis, where inflammation in the lung contributes to the disease, is that when you use these tricyclic antidepressants, in this case we focused on amitriptyline, one of the more potent ones, there was a significant reduction in ceramide in the lung. As expected, you're reducing this enzyme, so you see less ceramide. But there also was a significant reduction in the expression of, once again, interleukin-1 beta in the lung, as well as improved lung function. So we grabbed that molecule and wanted to see if we could see changes in microparticle production and inflammatory cytokine production. So our first test was in vitro. We took human sickled RBCs from our donors, threw them in a dish, and treated them with or without amitriptyline. We used flow cytometry to measure the production of microparticles and found that in a dose-dependent fashion, with the two highest doses having significant changes, there was a reduction in microparticle formation from these red blood cells sitting in dishes. We wanted to go back to our physiological mouse model, so we first looked at acid sphingomyelinase activity and found that once again, 24 hours after injection in a dose-dependent fashion, there was a significant reduction of acid sphingomyelinase activity at 10 milligrams of the drug per kilogram weight of the mouse. So we used that dosage and went back to our mouse model to see if we could detect changes in microparticle production. We found that microparticles themselves and erythrocyte-derived microparticles, or red blood cell microparticles, which had glycophorin A, which is a red blood cell marker, were able to be detected in the blood from these mice. And as expected, hypoxia induced significant increases in microparticle production, both microparticles and erythrocyte-derived microparticles. But amitriptyline reversed that trend. So with amitriptyline, you don't see a significant increase in microparticle production. That's great, right? We're not seeing microparticles, but do we also see changes in inflammatory, in systemic inflammation? Once again, we went back to our cytokines. We measured IL-6, TNF-alpha, and interleukin beta. So the results here were interesting. We didn't see a significant increase in cytokine expression in the blood of these mice induced by hypoxia, but even during normoxic or regular oxygen concentration conditions, we saw a significant reduction in IL-6 and interleukin beta. We previously shown that all of these cytokines are produced by monocytes, and to this point, we hadn't looked at neutrophils. And it's a different type of inflammatory cell that's also shown to be activated in sickle cell disease. The classic marker for neutrophil activation is myeloperoxidase, or MPO. So we got an ELISA against that protein and found that, though not significantly, there was an increase in MPO expression in the blood during hypoxia, which was reversed with amitriptyline. So we're not only seeing effects of inflammation mediated by monocytes, we're seeing markers of neutrophil activation also reduced. Lastly, we wanted inflammation in the lung was altered at all in the presence of amitriptyline. So acute chest syndrome is a complication that exists in many people living with sickle cell disease. Basically occurs when you have significant recruitment or infiltration of inflammatory cells to your lung vasculature, and you have vaso-occlusion in the lung. So we took a careful look at the cross-sectional slices of the lung, and we looked at vessels, and we saw a slight increase in the recruitment of inflammatory cells during hypoxia. And we haven't quantified this, but there seems to be a reduction of inflammatory cell recruitment to vessels in the presence of amitriptyline during hypoxia, suggesting that we're not just seeing changes in protein expression, we might be seeing changes in inflammatory cell recruitment in the mouse model of sickle cell disease. So altogether, AIM-1 showed us that sphingolipid metabolism is significantly dysregulated in sickle RBCs, and it leads to an altered steady state with activation of one particular enzyme, acid sphingomyelinase, which produces membrane-derived microparticles which contain sphingolipids. These sphingolipid-rich microparticles are internalized by myeloid cells, monocytes, and macrophages, and enhance monocyte adhesion, pro-inflammatory cytokine production, and genetic modification. In aim 3, we showed that we could pharmacologically reduce acid sphingomyelinase activity with amitriptyline and reduce microparticle production as well as vascular inflammation in a mouse model of sickle cell disease. I'm sure you guys are tired of the slide yet, but I put a lot of time into it, so I'm going to show you. So altogether, we know that sickling in sickle RBCs induces activation of acid sphingomyelinase. This enzyme converts the most abundant sphingolipid, sphingomyelin, into ceramide, which is further converted into sphingosine and S1P, the bioactive lipid. Microparticles are produced in sickle RBCs, which contain these lipids, and they can be secreted from the RBCs, where they interact with monocytes in circulation and are internalized, which can distribute their sphingolipid contents to monocytes. S1P and other lipids can activate S1P receptors, which initiate downstream signaling cascades that result in the production and secretion of TNF-alpha, IL-6, and interleukin-1 beta, as well as the adhesion of these cells to epithelial cells. We found a molecule, amitriptyline, that can inhibit acid sphingomyelinase activity and remove or reduce a lot of these downstream markers of inflammation in sickle cell disease. Future directions. If there's one thing I learned during grad school, it's that the work is never done. And I really hope that this work isn't done. I'm really excited about some of the observations that we've made, and I really hope that they can continue to be strengthened in the future. The first thing that we plan on doing is continuing to model and optimize continue to optimize the model and make noteworthy predictions of changes in flux, as well as specific enzyme activity measured by the reaction constants in our model of sphingolipid metabolism. To do that, we need to first really understand all of the enzymes and reactions that exist in red blood cells. So completely profiling all the enzymes that exist in these cells and understanding which ones are there and which ones aren't will really help us to design a more optimized model, which can then produce exciting therapeutic insights. We also really want to follow up the ERK observation. This showed that not just in red blood cells, but in the microparticle byproducts, there's significant activation of ERK1/2. We want to see if this activation is actually signaling in microparticles, and if ERK itself can be delivered to monocytes and other cells once the microparticles interact. So just truly understanding how signaling in general and ERK in particular is playing a role in the activation of these cells is exciting. We also want to continue to characterize the role of cathepsins in the activation of monocytes. Again, these are powerful proteases that have been shown to play roles in vascular remodeling and on the endothelium. So we really want to understand how these cathepsins are induced in monocytes and whether or not they themselves can be transferred to endothelial cells and other cells in the disease state. So an interesting result that I didn't really touch on was the phenotypic specific response to these microparticles. Some of my earlier work focused on the existence of distinct subsets of monocytes. In this case, the M0, 1, and 2 macrophage or monocyte, which are kind of quiescent macrophages, inflammatory macrophages, and pro-regenerative or anti-inflammatory macrophages. So in this study, we basically took these three different phenotypes and incubated them with microparticles. And we found that you didn't see the same exact change or same exact trend in terms of the cytokines that were induced. For IL-6, for example, you can see that microparticles enhance the production and secretion of IL-6 significantly in M0 and M2 macrophages, but not so much in M1 macrophages. So the idea that there are different phenotypes of monocytes that interact with and respond to the microparticles differently, but also have unique effects in terms of vascular remodeling. I mentioned the patrolling monocyte in the previous work. Is just really, really fascinating. We want to understand if there's one particular phenotype of monocytes and macrophages that's producing a lot of the inflammatory changes that we're observing, and if we can carefully target that phenotype to not disrupt other things that naturally happen. Lastly, this work used a tricyclic antidepressant and a tryptamine to reduce acid sphingolipid activity. But this isn't specific to acid sphingolipids. This one molecule plays roles in the nitric oxide pathway, prostaglandin 2. So the fact that it's not specific means that there are definitely going to be off-site or off-target effects of the molecule. So we really want to, by finding or creating a molecule that specifically inhibits acid sphingolipid's activity, we're finding others that are more potent, therapeutically effective targets of enzymes in the sphingolipid metabolic pathway. Okay, that's the hard part. I definitely want to start by acknowledging the people that helped me do this science. It's been a long journey. It's taken me to three different institutions, UVA, Georgia Tech, and Emory. But I really got to start by thanking the Bochray Lab. You guys have been, great family, great home, and you've just been really helpful in creating an environment that allows for exciting observations in the area that we work in. So if you can't tell, this says Bochray down here. This is an old picture of the lab. We recently coined the name LEAR, which stands for Laboratory for Immunoregenerative Engineering. And our daddy's Ed, leader of the lab. So like Ed, I can probably go on and on about our relationship over the years, but I'll be brief and say thank you for your guidance. Talk a little bit about this particular picture. So if anyone that spent time in our lab probably knows how this went. This was likely a Friday afternoon at 4:30 p.m. When the only thing on my mind was heading to the gym and starting my weekend. And Ed would call me into the office and just have a casual conversation about the newest and most recent experimental finding that turned into a 2-hour experimental design session. So moments like this, I truly cherish throughout the Ph.D. And Ed has been an amazing scientific mentor, but also, just emotional and a mentor in other ways as well. In terms of the experiments, I know this might be hard to see, but the conversation generally went like this. What that bubble up there says is, I go into Ed's office and ask if he wanted me to run, or which experiment he wanted me to focus on out of a number. This could be 2 through 30. And anyone who spent time in lab knows that without failure, the answer is always, we haven't practiced this. Do them all. I mean, there's truly no brake pedal, there's no slowing down in Bochray Lab, and he consistently encourages us to get our hands dirty and do everything we can, which you probably can appreciate from my talk. I've kind of got my hands in a lot of different areas, but I've loved it. Ed, though, is not afraid to get his hands dirty. He really encourages us to get our hands dirty. He does his work in different ways. That's him in the back. This was while we were at UVA. Shane, you might remember this day. We planned a field day with all the labs at the Department of Biomedical Engineering at UVA. And Ed was part of our team. This is me congratulating him after our win. But our lab's just really fun, energetic, and if I wasn't surrounded by people that knew how to do compelling work but also have a good time, I would not be standing here today. So I appreciate you all. All right, so there are 2 particular lab members that I want to highlight. I love you all, but these 2 were specifically impactful in bringing me to where I am. First, Dr. Awojoodu. She joined the lab around the time that we transferred from UVA, and she was just really, really helpful in getting us up and running. At the time she had her PhD, she just really was the only one that understood how to run a lab and get things up and running. And that process was tough. It took a lot of time to get protocols written and get to the point where we were actually running experiments. But also Molly and I worked quite closely together on my first author publication, which also wouldn't have gotten published if it wasn't for you. So thank you so much for your help. And Nathan, this is actually the only picture that we have here. Nathan's he's like a Sphingolipid metabolic computational guru. Everything about Sphingolipid metabolism. I knew nothing 2 years ago, and I'm still learning every day from Nathan. So it's just been really great building a relationship with you and being able to, without planning, just catch up and discuss, results that we have and new areas of exploration. So thank you for always being willing to listen to my dumb questions and explain things in a way that I can understand. So the PhD process is not all about publishing, traveling, and exploration. so you can't cry. It's very much so. I had the opportunity to mentor some amazing students. Mike, you're not on this side, but Mike was my first undergrad at UVA on some of the microparticle remodeling work that Shane and I worked on. There are other undergrads I've worked on throughout the years, but these two in particular, Sylvia, who's in the back, and Alicia, who's in the back right, have just been amazing undergraduate students to me. And they both started about 2 years ago in lab, knowing nothing about sickle cell, nothing about sphingolipids, nothing about the work that we do. And their eagerness and ability to just step in there and understand and work with me has been it's been an inspiration. I mean, you guys have helped me do the work that I do. More importantly, they're not just in lab doing lab chores. They're actually contributing to the work. They're both co-authors on the blood paper that I mentioned, and they recently just came back from the National Biomedical Engineering Symposium, where they gave talks and posters on some of the work that I just presented. So you guys inspired me, and you're going to do great things in science and Jovene, Carter, and Amadou Ba in the back. All right, so these guys are not undergraduate researchers, they're high school researchers. So Dr. Platt and others here at Georgia Tech established the ENGAGE program, ENGAGES now. Some of the students are in the back, but it's a great program that basically allows underrepresented students from the inner city to come to Georgia Tech and do research. And I mean, it's the first time really exploring the college campus and doing research. I didn't have any opportunity to research in high school. These guys aren't bashful, they're not afraid to speak up in lab meeting, and in similar ways you guys have inspired me to keep my mind on what's important, and that's the science, the compelling nature of the work. So thank you. Tiffany's not with us. She's a former lab mate of ours. She graduated with her master's and now she's out west working. But she also was elemental in getting the lab up and running when we moved from UVA. And she's really like a lab mom to all of us. So all the ordering that we needed to get done, inspections, cleaning, she just kept the wheels rolling. Yeah, take more pictures.
Too busy working.
Profile pictures. I don't feel so bad. Take this from Facebook, but Phil's just been really, really great in many ways. I mentioned that the Platt Lab had a collaboration with the Civil Foundation of Georgia that allowed us to get all the samples that were required to make the observations that we made. So just being able to facilitate that relationship for us was awesome. But also, this guy's like a molecular biology genius. I mean, he knows everything about experimental design and making measurements in the molecular biology area. So I appreciate you for that and for always answering like anything my dumb questions. Thank you. And Dr. Lynch, he's also in pharmacology at the University of Virginia. He was on my master's committee, but also helped us to make a lot of the HPLC mass spec measurements that I presented on. Our committee, you guys are concentrated in this corner over here. I've been fortunate enough to work with 4 professors outside of ed that have been, amazing sources of support, encouragement, motivation, and thinking. 2 from there and 2 from afar. So Dr. Platt and Dr. Gibson, I can't count the number of times that I just walked into your offices and talked about malaria, sphingolipid remodeling, cardiovascular disease with Dr. Gibson, planning grants and papers. With Dr. Platt, I can think of, I'll never forget, I should say, one example of the kind of mentor that he is. So we wrote the blood paper together, and I took the draft to him. Actually, I emailed the draft to him. It was an electronic version, something like 20 pages. I said, hey, get it back to me when you can. I expected like comments on the abstract or something. He printed out the draft, 20 pages, called me into his office and handed back a hard copy with no white space. All the space that was white was filled with white red notes. And I mean, it just showed how he really cares about the work that we put out. And I really appreciate your critical feedback along the years. Dr. Barabino, you probably don't know this story, and I've gone back and forth between telling it or not because it's a creepy story. So before I knew you, I knew who you were. And Dr. Barabino was the former president of the Biomedical Engineering Society, and graduate chair here at Georgia Tech before she moved to become dean in New York. And she was getting recognized for some really prestigious award at BMES. It was like my 4th time going to a conference, and she was on the same flight that we were on. was traveling with my former lab mate, Cynthia. And I pointed her out and I said, hey, well, that's Dr. Barabino. She's doing the sickle cell work at Georgia Tech. Like, she's awesome. Look at her profile in the book. And she encouraged me to walk up to you and introduce myself and just say, hey. And at that time, I had no idea that Eddie would get recruited to Georgia Tech and the whole lab would be uprooted and moved down here. And she'd be on my committee years later. And, you know, she was open when I met her and smiled and shook my hand, but I never forgot that exchange. And I'm really fortunate to have you on my committee. Thank you. And Shane, who, are you still there, Shane?
Yep, I'm here on mute.
It's like the voice of God. Shane, she's probably one of the most hats on my committee. She was my teacher at one point at UVA. She was on my master's committee. She was on my comprehensive exam committee. She's my master's committee chair. She's a co-author. She's now on my Ph.D. committee. And you know, while I moved away from Georgia Tech and she's not physically here, her presence is felt, you know, more than anyone else I've been able to work with throughout this, in terms of committee members throughout this process. So I really appreciate you always being there. She never hesitates to pick up the phone and call about, you know, whatever is important or relevant to me at the time. And I think we built a great working relationship between the two labs that I hope can continue. So thank you. I'm almost done here. Funding sources. We couldn't do this work without funding, so we've had multiple NIH and NSF grants, so definitely have to acknowledge them, as well as the Sickle Foundation of Georgia for giving us the samples that we used. There are a lot of people here at Georgia Tech and at UVA that it take too long to mention. The support staff, the Shannon in the front office, all the cores that are here, you know, just so many systems that are set in place to allow us as grad students to do the work that we can do with as little pain as possible. So I'm really grateful for that, as well as the Black Graduate Student Association. Many of you are here. Thank you for being my outlet, my stress relief along the years. I appreciate it. Family, that's me. I'd first like to acknowledge and thank Mommy and Daddy. Can you guys bring? I think you're wearing the same hat now. First and foremost for bringing me into the world, but also for, I mean, this guy flew from Nigeria to be here and just always being supportive for me. My mom is the most hardworking, strongest person I know. You've always set an example that I can look up to. I just hope I can raise my kids half as good as you raised us. So thank you. My little sister, you have to wait. My baby sister, she's a grown adult now. She actually has sickle cell disease. She's 21. She's lived with it for 21 years, and she's just a warrior. I mean, she's never used it as an excuse, you know, through pain, through hospitalization, anything that it's, you know, any hurdle that's created. She's never used it as an excuse to not be great, and you know, not smile and do the work that she needs to. And you know, your outlook on life is one that everyone needs to have. And though you're my baby sister, you're an inspiration. I love you. The rest of my siblings, Richie, Joycey, Faith, and Bella, for putting up with me for a lot longer than they deserve to. Support systems as well as my brother-in-law and my two nephews who think that it's too cool to smile with me. My in-laws, Shoa's aunt, Shoa's my fiance, my aunt just left, she was here earlier, but none of them can make it. But that's an example of them always wanting to feel, you know, present in my accomplishments. So I appreciate them for supporting me as if I was, you know, in their family, but also for giving me my best friend, fiance and wife to be, Shoa. So this is taken back in 2006. So she's had multiple opportunities to dump me or but as you can see, a lot's changed over the years, but two things that have stayed the same are my affinity to gray suits and just her love and support. And she couldn't be here today because she's out there saving lives. She's rotating in med school right now. But I'm excited to spend the rest of my life with her and she's been awesome. All right, fortune. I've been fortunate enough to participate in so many different experiences, both at UVA, here at Georgia Tech and Emory, and throughout the world. I'll speak to three because I know you guys are probably hungry and tired at this point. So Ed was crazy enough in 2010 to let me go to my first international conference. It's called the Ghana Biomedical Conference. And yes, the story that he told, he's the Ghanaian, I'm the Nigerian. He built this community of friends and collaborators there that really wanted to push biomedical engineering to the forefront in Ghana, and it was a great opportunity for me to go and represent the lab, speak and meet people working in an underdeveloped nation, and really see how science and research operated there. Next year, I did an externship. I was a visiting scientist for AstraZeneca, which is a pretty big pharmaceutical company. I was in Sweden, which was awesome. This is my PI Shin Dong, working on stem cell mobilization for cardiac regeneration, and got the opportunity to explore the ins and outs of research and development in the pharmaceutical industry, and the ins and outs of Europe, which was kind of cool too. Lastly, most recently, Ed again was crazy enough to let me leave for an internship in the 11th hour of my thesis. My former internship company and future employer, McKinsey Company. So I was there working again in the pharmaceutical area on drug regulatory issues. And while there, I just got the opportunity to meet and interact with some of the most brilliant, passionate people that I've ever encountered. One of which is this guy, Cory Booker. He's awesome. Hopefully he'll be president someday. But these experiences have really shaped my graduate career in a positive way, and I'm very appreciative for all the open doors over there. And lastly, how that can signal. In terms of what the lipids are doing, the immunofluorescence imaging that shows the confocal videos show that the lipid contents or the intracellular contents of the microparticles can be distributed throughout the entire cell. So I don't believe that all the contents just sit in the membrane. The lipids can be internalized and processed in the endoplasmic reticulum or Golgi apparatus, as well as other proteins in there. So I mean, I think it'd be really interesting to look at what else is there outside of lipids. But before we could say what exactly is mediating some of the changes, we have to know what's in these microparticles.
Do you think they're being actively targeted to macrophages or monocytes, or can they interact with the endothelium or any other?
Yeah, so I think both are possible. So they could just be endocytosed, right? And we haven't explored this yet, so we can't say for sure. And that wouldn't be a very active targeting. And any phagocytic cell could internalize the particle. But the result that I think is one of the more interesting ones in our work is that ICAM4 ERK phosphorylation story. So we know that red blood cells that are sickled have ERK1/2. It's activated. Downstream of that activation, they activate ICAM4, which is intracellular adhesion molecule 4. And that's the receptor that actually binds to monocytes. So the monocytes have the substrate for that ligand, or the ligand for that receptor, and that's what mediates the contact. The microparticles have phosphorylated ERK. They also have ICAM4. So the target could be with ICAM4 or other adhesion molecules. And again, that's not necessarily specific to monocytes, but it's specific to any cell that has a ligand for ICAM4.
Yeah, great job, Tony. Very proud of you. Mine is a layman's question, very simple. With this work, what does the future hold for sickle cell patients?
Great question. I mean, I think a lot of work now is really trying to understand, from a genetic basis, how we can target the disease genetically. And I think in the future, genetic engineering in terms of the cure of sickle cell disease might be a potential option for humans. But right now we're kind of limited to treating these hydroxyurea, transfusions are ways that patients normally to treat the disease and the complications of it. But one of the first slides I mentioned shows the financial aspects of the disease. Those treatments generally require hospitalization. It's very costly. And they don't really change the disease state. They help you deal with pain or get fresh red blood cells for a certain amount of time. So I think the future of this work is finding therapies, whether it be mediated by acid sphingolipid or something else, that could exist in a pill or an injection. And someone living with the disease could take a medicine that would modulate the enzymes instead of just trying to replace the cells, so that the complications of the disease would be reduced kind of consistently throughout their life. And they wouldn't have to worry about the more costly remedies such as transfusion. Can I grab a really quick? Okay.
My question is dealing with the future directions that you explained with sphingolipid. You said that one of the future directions could be to create a drug or molecule that blocks sphingolipid. Do you have any ideas or theories of what that drug could entail?
I don't. So at one point we were talking with the lab, and I mean, this is the beauty of being here at Georgia Tech. This is IBB, which has multiple different disciplines and departments. So we used to sit up in a wing with some chemists that were really interested in the work that we did. And they said, hey, if you just give us the structure of the molecule, we can develop an antagonist or inhibitor for it. So a lot of us, maybe Nathan, but outside of Nathan, none of us have a real strong background in chemistry or synthesis. But I think it's possible to collaborate with groups that can design molecules that specifically target that.
Yeah, I think your dad had the most brilliant question and follow up on that, because I come from more of the translational side, and similar to the question we just asked about where the targets are. I think this is, Rob, I just want to commend you. It was a brilliant, I think it was brilliant work, brilliant presentation. I had seen that JCI paper, and then I came and spoke with Dr. Bertrand about it, and he explained, part of what you're doing. So I understand that the systemic aspect of the microparticle, but I also kind of wanted to go back inside the red blood cell itself, because I tried to get my head around this. I'm not, a card-carrying scientist, but it seems to me that there's almost a chicken egg between like the SPHK and the actual sickling. So I was just wondering, how far do you think you can get toward reducing the sickling process itself, as opposed to then, having to deal with the microparticles that are going around acting as inflammatory vectors by going after these SPHK or SMAs? I mean, do you think that there's a minimum amount of this sort of sphingolipid activity that needs to go on to actually facilitate the sickling? Or do you think the sickling will always be there and it's just a feedback that like cycles it up?
Yeah, that's a really interesting question, and I mean the best way to describe it is exactly what you said, chicken egg. I mean, the way that I understood most of my work before we looked at that paper was that it's the sickling in the red blood cell membrane that activates the enzyme, acid-sphingolipid, that produces these microparticles and changes the lipid state. But in the paper, it's kind of the other ways. It's changes in the lipid profile that can contribute to sickling. So, I mean, I think it's an intriguing question. I don't know which one dominates. I don't know if they both kind of work together and there's a balance. But we do have a collaborator, at UVA, that was really interested in sphinxin kinase 1, not 2, inhibition in the area of sickle cell disease. I think the one issue, and this isn't unique to that inhibition, it's also going to play a role in looking at sphingolipid, is that these enzymes exist in all cells, and what's unique about them in the context of red blood cells is that the red blood cells don't produce them. So there are other cells, endothelial cells, monocytes, that produce these enzymes and potentially dump them out into plasma, and they're adopted by red blood cells. So even if we find out definitively that sphinxin kinase 2 or sphingomyelinase is upstream of sickling, we still have to design a therapy that reduces the enzyme's secretion into plasma, because targeting the red blood cells is only going to target what's there at that time. And if it's still being produced and secreted by other cells, then we still see the same response, or we might see the same response. But I mean, I think it's an interesting area, and hopefully someday we'll really understand what's going on.
Thanks.
All right, maybe we'll let that be the last question. So thank you for everyone. I move to our private session where we drill Tony.
So, all right. I'm gonna take your cut off because it died.
The laptop?
Yeah. It died right when you said you finished your acknowledgments.
That works.
So do you want me to take your hands to
Yeah, just somewhere else.
Okay. I'll take them across to the other building.